Ticks. Many people immediately think of them as bloodthirsty parasites. But little do people know that these harmful creatures are responsible for an even more deadly infection called Lyme disease. Lyme disease is a vector-borne illness caused by the pathogenic bacterium known as Borrelia burgdorferi, a spirochete transmitted from a black-legged tick bite. Tick-borne spirochete infection is dependent on a triangle of interactions between the burgdorferi, the reservoir vertebrate host, and the tick vector. The tick usually acquires the pathogenic spirochete from feeding on the reservoir vertebrate host, most commonly the white-footed mouse. The tick feeds by plunging its mouth parts into the host's skin and feeding on the host's blood. This feeding method makes the tick susceptible to pathogenic infection. The spirochete infection can be traced through the first two phases of the tick life cycle. The full life cycle of the black-legged tick spans over 2 years and comprises 3 phases: larvae, nymph, and adult. The tick requires a blood meal to pass from one life stage to the next. This can be broken down into 3 significant meals over its lifespan. The first meal occurs when host-seeking larvae that are not yet infected with the spirochete enter their peak feeding activity in August. This is where most vector infection occurs. The larvae molt into the nymph life stage and go dormant until early spring. The second meal occurs when the nymph's peak feeding activity occurs during the late spring through midsummer, when most people are enjoying the outdoors. Ticks in the nymph stage are the primary transmitters of the pathogen. The nymphs are small enough that they go unnoticed while feeding, allowing plenty of time to transmit the spirochete into the bloodstream. The bacterium thrives within the human body, waging a full-on attack. The body's immune system does little to inhibit the spread of the bacterium, causing the body to undergo 3 different phases in response to the disease. Phase 1 of the disease includes a localized infection presenting a bullseye-shaped rash around the bite wound. This leads to Phase 2, where the infection has spread throughout the body. This presents complications such as joint pain, general inflammation, persistent arthritis, and neurological symptoms. Phase 3, when the infection has spread into the brain, occurs only when the infection goes untreated with antibiotics, consequently creating paralysis, problems with speech, memory loss, vision impairment, and other neurological damage. The first documented cases of Lyme disease were in 1975 in the small rural town of Lyme, Connecticut. The black-legged tick is the most common vector for Lyme disease in the United States. Vectors are defined as the transporters of a pathogenic organism from one host to another. One of the key elements in the effective spread of vector-borne illness is the environment that the host and vector share. Borrelia burgdorferi can infect a multitude of small mammalian and avian species. However, the white-footed mouse is the primary reservoir host carrying the pathogen in the northern regions of North America, where the spread of Lyme disease is most prevalent. White-footed mice are responsible for the infection of 40 to 90% of the immature and mature tick population. White-tailed deer act as the primary host, responsible for transmitting the infected ticks from one animal to another. The deer are the pivotal step between the reservoir host carrying the pathogen to human infection. The reforestation of farmland in the northeast region of the United States over the past 30 years has given way to the repopulation of white-tailed deer. Over the last 20 years, the black-legged tick has slowly expanded its population from eastern North America to include now the central and northwest regions of North America. One study shows that biodiversity decreases the occurrences of Lyme disease. Larger and more diverse populations of animals leave little room for the preferred host's population. This in turn keeps the vector population down. Lyme disease was first notable and surveyed in 1991, and since then annual case reporting have doubled. In 2000, there were 18,000 cases of Lyme disease reported, and since then there have been over 20,000 new cases of Lyme disease reported each year in North America. Warmer and wetter climate change over the last 10 years have led to tick population growth and survival through colder seasons when the populations would traditionally deplete. Lyme disease can only be spread to humans when hosts, vectors, and humans are within a close proximity of one another. The warmer climate change fosters more outdoor activity in communities around New England, thus extending the general tick season and human exposure to infected ticks. Tick season is defined state by state. For example, the state of Massachusetts references the summer months as tick season. Through climate change, longer exposure to larger host and vector populations will inevitably lead to an abundant growth in Lyme disease infections. The effect of reforestation and climate change on Lyme disease are clear examples to just how dramatic the effect of ecology is on vector-borne illnesses.